Hi, my name is Meytal Landau, and I want to tell you about fascinating proteins that we study. And proteins in general, like the ones that we eat or that are composed or that compose our body, are super tiny. You can fit many thousands of them across one strand of a human hair. And many of them, maybe most of them, they have a defined three-dimensional structure like a bulb. But we study unique proteins that are called amyloids that assemble into very strong and long fibers. You cannot still see them by eye, but they are longer even from a whole cell of bacteria. And these amyloids are mostly known because you find them in the brains of people suffering from Alzheimer's and Parkinson's and other diseases. But we now know they are basically everywhere. For example, microbes like bacteria and fungi and even viruses produce amyloids for their own benefit. For example, they can make the infections much more aggressive. They attack our immune system and they colonize surfaces. And now we also know, we found that some of the amyloids that are produced by creatures like frogs and insects and humans, they form these amyloids in order to serve as immune defense to protect us. So we asked the questions, what makes amyloids sometimes very beneficial and sometimes very harmful? Is there, and what is the connection between microbes and their amyloids and neurodegenerative diseases? And importantly, how can we control them? So to answer these questions, we want to look at amyloid structures and in different environments in very, very fine details. And to do this, we use very powerful X-ray beam at the Petri synchrotron here in DESY, and also state-of-the-art cryogenic electron microscope at the CSSB Center for Structural System Biology, where we are here now. And we want to use this information that we get here in order to design therapeutics and other applications.